Evo2, the largest open-source AI model for biology. Release date, February 19, 2025. Overview. Evo2 marks a transformative advancement in the field of generative biology developed through a collaborative effort among the Arc Institute, NVIDIA, and prominent academic institutions including Stanford University, UC Berkeley, and UC San Francisco. It is recognized as the largest open-source AI model for biology trained on an extensive dataset comprising over 9.3 trillion nucleotides from more than 128,000 genomes across diverse lifeforms including bacteria, archaea, plants, and animals including humans. Key features and capabilities. 1. Pathogenic mutation prediction. Evo2's primary strength lies in its ability to predict pathogenic mutations in human DNA with over 90% accuracy. This was highlighted through tests on the BRCA1 gene, which is linked to breast cancer. By identifying genetic causes of diseases more efficiently, Evo2 has the potential to significantly expedite research processes. 2. Genome design. Beyond mutation prediction, Evo2 can design novel genetic sequences. This capability opens avenues for targeted gene therapies, allowing researchers to create genetic elements that activate in specific cell types, thus reducing side effects and enhancing treatment precision. 3. Generative biology. Evo2 represents a leap toward understanding and manipulating genetic information akin to how large language models process textual data. By discerning patterns in nucleotides, Evo2 can read and write genetic code, effectively speeding up evolutionary processes and enabling the design of new genetic pathways. Technical specifications. The development of Evo2 was a monumental endeavor involving over 2,000 NVIDIA H100 GPUs and leveraging the NVIDIA DGX Cloud AI platform. The model utilizes a unique architecture called StripedHyena 2. Which allows it to process genetic sequences of up to 1 million nucleotides simultaneously. This capability is crucial for analyzing complex genomic interactions that shorter context models often overlook. The training process took several months and aimed to overcome limitations seen in its predecessor Evo1, which focused solely on single-cell genomes. Ethical considerations. In developing Evo2, the team prioritized ethical considerations by excluding genomes from pathogens that infect humans and complex organisms. This decision aims to minimize risks of misuse while ensuring that the model serves beneficial purposes in research and medicine. The developers engaged experts to guide the responsible use and deployment of the technology. Collaborative development. Evo2 emerged from a partnership between multidisciplinary teams combining machine learning specialists, computational biologists, and experimental researchers. This collaborative approach ensured the model's effectiveness and utility in real-world applications. The project highlights the importance of teamwork in tackling complex scientific challenges. Potential impact on research and medicine. Evo2's capabilities extend to various domains including accelerated drug development. By identifying genetic causes of diseases, Evo2 could streamline the process of drug discovery, potentially saving researchers time and resources. Bioengineering applications. The model's ability to design new genetic sequences could lead to innovative biological tools and therapies addressing both health and environmental challenges. Integration with experimental biology. Researchers can leverage Evo2 to enhance experimental designs and hypotheses, paving the way for novel discoveries in genetics and molecular biology. Timeline of key events. February 19, 2025, Evo2 was officially released accompanied by a user-friendly interface called Evo Designer. The model's code, training data, and other resources are made publicly available on GitHub. Preceding development, Evo1 laid the groundwork for Evo2, demonstrating the feasibility of AI in genomic analysis. Collaborative training. The training of Evo2 involved significant computational resources and reflected a large-scale collaborative effort among multiple research institutions. Conclusion. EVO2 stands at the forefront of biological research, offering researchers an unprecedented tool for genomic analysis and design. By advancing our understanding of genetic information and its implications for health, EVO2 is poised to foster innovation in medicine and bioengineering while adhering to ethical standards. The release of this fully open-source AI model represents a significant milestone in the intersection of artificial intelligence and biology, setting the stage for further advancements and applications in the scientific community. For more detailed insights, please refer to the preprint Genome Modeling and Design Across All Domains of Life with EVO2, available on bioRxiv. Along with the companion machine learning papers, Systems and Algorithms for Convolutional Multi-Hybrid Language Models at Scale. The EVO2 AI model, touted as the largest open-source biological AI model to date, presents several counterintuitive aspects that challenge conventional wisdom in genetic research and the application of artificial intelligence in biology. 1. Open source versus security risks. Traditionally, the release of powerful AI models raises concerns about potential misuse, especially in sensitive fields like genetics. However, EVO2's developers have chosen to make it fully open source while implementing restrictions on its ability to generate harmful biological agents. This move subverts the expectation that powerful technology should be tightly controlled or kept proprietary to prevent misuse. Instead, they opted for transparency and collaboration, fostering an environment where researchers can innovate while adhering to ethical standards. 2. Speeding up evolution. The idea of an AI model being able to speed up evolution is counterintuitive because evolution is typically a slow, gradual process that occurs over millions of years. EVO2, however, allows for rapid predictions and the generation of genetic sequences, potentially mimicking evolutionary processes in a fraction of the time. This capability to quickly simulate and test genetic changes might lead to advancements in understanding complex traits and diseases in ways that traditional methods could not achieve. 3. Generalist versus specialist models. EVO2 operates as a generalist model that can address a wide range of biological questions, from predicting disease-causing mutations to designing new genetic sequences. This challenges the prevailing notion that AI in biology should be highly specialized, focusing on narrow tasks. Instead, EVO2's architecture allows it to generalize across various domains of life, which could lead to unexpected and innovative applications that researchers have yet to fully envision. 4. Predictive accuracy versus traditional methods. With more than 90% accuracy in predicting pathogenic mutations, EVO2's performance challenges the belief that computational methods cannot match or surpass traditional experimental approaches. This model significantly reduces the time and resources typically required for experimental validation in genetic research, suggesting that AI can not only complement but potentially replace certain experimental methodologies. 5. Integration of diverse biological data. EVO2's dataset includes genetic information from an extensive array of organisms, including bacteria, archaea, plants, and animals, which is often seen as a limitation in models focusing on specific organisms. The counterintuitive element here is that by integrating such diversity, EVO2 can uncover patterns and relationships that might be completely overlooked when focusing solely on a single species or type of organism. This broad scope challenges the typical reductionist approach in biology, advocating for a more holistic view of genetic interactions. 6. AI as a collaborative partner. EVO2's development involved collaboration across multiple prestigious institutions, which is a contrast to the perception of AI as an independent entity that operates in isolation. This collaborative effort highlights the importance of interdisciplinary work in advancing scientific knowledge, suggesting that AI can serve as a partner to human researchers, enhancing rather than replacing their efforts. 7. Ethical considerations in AI development. The developers of EVO2 have proactively excluded genomes from pathogens that infect humans, challenging the assumption that powerful AI tools are inherently dangerous and should be restricted. Instead, they have taken steps to ensure that the model's capabilities do not lead to negative outcomes, demonstrating a commitment to ethical AI development that contrasts with the often pessimistic views surrounding AI in sensitive areas like biology. 8. Data utilization. The sheer scale of EVO2's training data, consisting of over 9.3 trillion nucleotides, raises questions about the efficiency of data usage in AI models. One might assume that larger datasets inherently lead to better performance, but EVO2's architecture and training methods demonstrate that how data is processed and utilized is just as crucial as the quantity. This insight challenges the notion that more data is always better without considering the underlying model architecture and training strategies. In summary, EVO2 embodies several counterintuitive ideas that challenge established beliefs in the fields of genetics and artificial intelligence. From its open-source nature to its ability to accelerate evolutionary processes, EVO2 presents a new paradigm for understanding and manipulating biological systems, suggesting that the integration of AI into biology may yield innovative solutions to complex challenges. EVO2, the latest AI model for biology, is a remarkable achievement in the realm of generative biology, representing a major leap forward from its predecessor EVO1. While many reports focus on its capabilities to predict pathogenic mutations and design new genomes, several important aspects of EVO2 are likely to be overlooked by the general public, yet hold significant implications for the future of medical research, bioengineering, and ethical considerations in AI applications. 1. Collaborative nature of development. One of the standout features of EVO2 is the collaborative effort behind its development. The project brought together the Arc Institute, NVIDIA, and leading academic institutions like Stanford University, UC Berkeley, and UC San Francisco. This interdisciplinary collaboration not only highlights the complexity of the project but also underscores the importance of pooling resources and expertise in tackling significant scientific challenges. The collaborative aspect is significant because it sets a precedent for future research endeavors that require diverse expertise. By demonstrating how collective efforts can yield groundbreaking tools, EVO may second inspire similar collaborations across different fields, leading to accelerated advancements in science and technology. 2. Ethical considerations and safety protocols. EVO2's developers took notable precautions regarding ethical implications. They consciously excluded genomes from pathogens that infect humans or complex organisms from the training dataset. This ethical foresight is vital in the context of AI applications in biology, where the potential for misuse could lead to harmful outcomes. This aspect is significant as it reflects a growing awareness within the scientific community about the responsibility that comes with powerful technological advancements. As AI continues to permeate various fields, setting ethical standards and implementing safety protocols will be essential to prevent misuse and ensure that technology benefits humanity. This proactive approach can serve as a model for other AI developments. 3. Open source accessibility. EVO2's release as a fully open-source model with publicly accessible code and training data is another critical detail that may be overlooked. This transparency fosters collaboration and innovation, allowing researchers around the world to build upon the work done by the EVO team. By making the model readily available, the developers promote an inclusive approach to scientific research, where even those with limited resources can leverage advanced tools. The significance of open-source models lies in their potential to democratize access to cutting-edge technology. By lowering barriers to entry, EVO may second empower a broader range of researchers, particularly in developing countries, to contribute to advancements in biology and medicine. This democratization could lead to diverse applications and innovations that might otherwise not emerge in a more closed system. 4. Integration with existing frameworks. EVO2 is integrated into the NVIDIA BioNeMo framework, which aims to enhance scientific discovery. This integration is crucial as it allows for the seamless incorporation of EVO2 into existing research workflows and tools, thereby maximizing its utility. Understanding the technical ecosystem around EVO2 is important because it highlights how advancements in AI can complement and enhance traditional scientific methods. Researchers can leverage EVO2 alongside other tools, facilitating a more holistic approach to genomic research. This integration can accelerate the pace of discovery and innovation, leading to faster advancements in medicine and biotechnology. 5. Potential for future applications. While many reports emphasize EVO2's capabilities in mutation prediction and genome design, the broader potential for future applications in various fields is often understated. The model's versatility can extend beyond medical research into areas such as agriculture, environmental science, and synthetic biology. For instance, EVO2 could be used to engineer crops with improved resistance to pests or diseases, thereby contributing to food security. The significance of this potential lies in the model's ability to address pressing global challenges. As the world faces issues like climate change and food shortages, tools like EVO2 can play a pivotal role in developing innovative solutions that can benefit society at large. 6. Technical advancements in AI architecture. The architectural innovations behind EVO2, particularly the StripedHyena 2 architecture, are noteworthy yet often glossed over. This new architecture allows the model to process genetic sequences of up to 1 million nucleotides at once, a substantial increase in capacity compared to previous models. The significance of these technical advancements cannot be overstated. By enabling the analysis of longer genomic sequences, EVO2 can uncover complex genomic interactions that were previously difficult to study. This capability may lead to new insights into gene functions and interactions, ultimately enhancing our understanding of biological systems. 7. Mechanistic interpretability. The development of a mechanistic interpretability visualizer created in collaboration with the AI research lab Goodfire is an important feature that is likely to be overlooked. This tool allows researchers to understand the key biological features and patterns that EVO2 learns to recognize in genomic sequences. The significance of interpretability in AI models, especially in highly complex fields like biology, is profound. As AI becomes more integrated into scientific research, understanding how models make predictions and decisions will be essential for validating their outputs and ensuring their reliability. The focus on interpretability reflects a growing trend in AI to foster trust and accountability, which is crucial for broader acceptance and application in sensitive areas like healthcare. Conclusion. EVO2 is a landmark achievement in the field of generative biology, and while its capabilities in predicting mutations and designing genomes are impressive, the multifaceted implications of its development are equally important. The collaborative effort, ethical considerations, open source accessibility, integration with existing frameworks, potential future applications, architectural advancements, and focus on mechanistic interpretability are all significant elements that merit attention. As the scientific community continues to explore the capabilities of EVO2, it is essential to recognize the broader implications of this technology. By doing so, researchers can ensure that advancements in AI and biology are leveraged responsibly and effectively to address some of the most pressing challenges facing humanity today. 1. Revolutionary predictive capabilities. EVO2's primary strength lies in its ability to predict pathogenic mutations in human DNA with over 90% accuracy, particularly demonstrated through its analysis of the BRCA1 gene associated with breast cancer. This capability enables researchers to identify genetic causes of diseases more rapidly and efficiently, potentially expediting the drug development process. The model's proficiency in distinguishing benign mutations from those that could lead to serious health conditions marks a significant advancement in genomics and personalized medicine. 2. Genomic design and bioengineering applications. Beyond mere prediction, EVO2 can design new genetic sequences, offering groundbreaking potential for gene therapies. By enabling targeted activation of genetic elements in specific cell types, EVO2 minimizes side effects and enhances the efficacy of treatments. This capability not only paves the way for innovative medical therapies but also facilitates the creation of novel biological tools that can address various environmental and health-related challenges. The implications for bioengineering underscore EVO2's versatility as a foundational tool for future research and applications. 3. Collaborative and ethical development. The development of EVO2 is a product of extensive collaboration among renowned institutions such as the ARC Institute, NVIDIA, Stanford University, UC Berkeley, and UC San Francisco. This collaborative effort highlights the importance of interdisciplinary approaches in tackling complex scientific challenges. Additionally, the developers have taken significant steps to ensure ethical deployment by excluding genomes from pathogens that infect humans, showcasing a commitment to responsible research and minimizing risks associated with AI in biology. For technical innovation and scalability, EVO2 represents a leap in AI architecture and computational power, utilizing over 2,000 NVIDIA H100 GPUs to process genomic data comprehensively. The new architecture StripedHyena 2 allows the model to analyze genetic sequences of up to 1 million nucleotides at once, significantly expanding its analytical capabilities compared to its predecessor EVO1. This scalability not only enhances the model's ability to uncover complex genomic interactions but also sets a precedent for future advancements in generative biology, reinforcing the model's role as a cornerstone for specialized AI applications in the life sciences. 1. The Human Genome Project, 1990 to 2003. This landmark scientific endeavor aimed to map all the genes in the human genome, consisting of over 3 billion base pairs. The project was an international collaboration involving multiple institutions and countries, ultimately resulting in a complete reference sequence of human DNA. The knowledge gained from the Human Genome Project has had profound implications for medicine, genetics, and biology, leading to advances in personalized medicine, genetic testing, and our understanding of genetic diseases. 2. CRISPR-Cas9 gene editing technology, 2012. The introduction of CRISPR-Cas9 as a gene editing tool revolutionized genetic engineering by allowing precise modifications to DNA. Developed by scientists such as Jennifer Doudna and Emmanuel Charpentier, CRISPR-Cas9 enables researchers to edit genes in organisms quickly and accurately. This technology has vast applications from agriculture to medicine, including potential treatments for genetic disorders, cancer, and infectious diseases. 3. Synthetic biology and the creation of synthetic life, 2010. The J. Craig Venter Institute created the first synthetic bacterial cell known as Mycoplasma mycoides JCVI Syn 1.0 by synthesizing its entire genome from scratch and inserting it into a bacterial host. This groundbreaking achievement in synthetic biology demonstrated the potential to design and create new forms of life, which could lead to advancements in biotechnology applications including biofuels, pharmaceuticals, and environmental remediation. EVO2, the largest open-source AI model for biology, is set to transform our understanding of genomics and its applications in medicine and bioengineering. Released on February 19, 2025, EVO2 represents a significant leap forward from its predecessor EVO1 and is poised to influence numerous fields within biology. Here's a look into the future outlook surrounding EVO2 and its potential implications. Predictions for the future of EVO2. 1. Accelerated drug discovery and personalized medicine. EVO2's ability to accurately predict pathogenic mutations could revolutionize drug development processes. By identifying genetic variations that lead to diseases with over 90% accuracy, researchers can shorten the timeline for identifying therapeutic targets. This capability will facilitate the creation of personalized medicine approaches tailored to the genetic profiles of individuals, allowing for more effective and targeted treatments. 2. Enhanced gene therapy techniques. The model's capacity to design precise genetic sequences could lead to advancements in gene therapy. Researchers may develop therapies that activate or deactivate genes only in specific tissues, minimizing side effects and improving treatment efficacy. This precision could usher in a new era of gene therapy where treatments can be customized based on a patient's unique genetic makeup. 3. Integration with other AI models. As EVO2 is seen as a foundational model, we anticipate the emergence of specialized AI applications built on top of its architecture. These models could focus on specific biological challenges such as predicting protein folding, understanding complex gene interactions, or optimizing metabolic pathways in synthetic biology. The collaborative nature of EVO2's development will likely inspire further interdisciplinary research efforts. For environmental and agricultural applications, beyond human health, EVO2 could be applied to environmental science and agriculture. By designing genomes for crops that are more resilient to climate change or disease, researchers could enhance food security. Similarly, EVO may facilitate the engineering of microorganisms capable of bioremediation, effectively addressing pollution and waste management challenges. 5. The ethical considerations and regulations. As EVO2 allows for unprecedented manipulation of genetic material, ethical considerations will inevitably arise. Future discussions surrounding the use of AI in biology will focus on responsible practices to ensure safety and mitigate risks associated with gene editing. Regulatory bodies may develop frameworks to oversee the applications of EVO2 to prevent misuse and ensure ethical compliance in research and clinical settings. 6. Public engagement and understanding. As the capabilities of EVO2 become more widely recognized, there will be an increased emphasis on public engagement in genetic research. Educational initiatives will be critical for demystifying AI's role in biology and fostering a better understanding of genetics among the general population. This engagement will be essential in shaping public perception and acceptance of genetic technologies. 7. Collaboration across disciplines. EVO2's development underscores the importance of interdisciplinary collaboration. Future research will likely involve partnerships between computational biologists, AI specialists, geneticists, and ethicists, fostering a holistic approach to addressing complex biological questions. This collaborative environment will enhance innovation and lead to breakthroughs that may not be possible within siloed disciplines. 8. Advancements in synthetic biology. The generative capabilities of EVO2 could significantly advance synthetic biology by allowing scientists to design organisms with entirely new functionalities. This could lead to the creation of synthetic microbes that produce biofuels, pharmaceuticals, or other valuable compounds, pushing the boundaries of traditional biological systems. 9. Global impact on health. With its open-source nature, EVO2 democratizes access to advanced AI tools in biology. Researchers worldwide, especially in developing countries, can leverage EVO2 for groundbreaking studies that contribute to global health initiatives. This accessibility can help address health disparities and enable innovative solutions to pressing health challenges. 10. Continuous learning and improvement. As more researchers utilize EVO2, its algorithms will benefit from continuous learning. Feedback from real-world applications will refine its predictive capabilities, leading to even greater accuracy and efficiency in genomic analysis and design. Conclusion: EVO2 is poised to be a transformative force in the biological sciences, with implications reaching far beyond the laboratory. Its capabilities in predicting mutations and designing genomes will accelerate advancements in medicine, agriculture, and environmental science. As the scientific community embraces this groundbreaking tool, it will be crucial to address the ethical and regulatory challenges that arise, ensuring that the potential of EVO2 is harnessed responsibly for the benefit of humanity. The future of biology empowered by AI promises to be a landscape of incredible possibilities, innovation, and discovery.